So for today's HALS call, I wanted to give you guys an update on Bio2RDF, which we're calling Release 2. And this is work that's primarily been done really only in the last, let's say, half year or so. After my sabbatical, when I went back to Ottawa, I decided, along with my two students, Allison Callahan and Jose Cruz Toledo, to basically reinvest ourselves in Bio2RDF and basically try to consolidate the project and improve its overall quality and performance. And you can imagine that this is going to be a long-term project, and there are many things that we know we have to do, but we're slowly sort of chipping away at the issues and trying to make progress. And hopefully, as both the quality and the sort of response time for our resources improve, then I think there'll be more opportunity for you guys, whether you're users or whether you're software developers and you want to demo your applications with linked data in the life sciences, that this becomes a core resource that not only we maintain, but hopefully that you also can contribute to in some way. So let me basically start by saying that Bio2RDF is an open source project, and everything that we do is available online, and we're trying to establish, I think, better processes for both developing the resources that we have, but at the same time enabling other communities and groups and researchers to make contributions that they think will help improve this project. And so in that sense, we don't intend to fully control the development process, but we want to engage with other people to find that better place that we have to be at. So basically, the hope here is that Bio2RDF will be something that makes the vast amounts of biological data that's available and make it available on this emerging semantic web. And so we've already seen the growth of the linked open data cloud. There's a substantial portion which does come from Bio2RDF and the life sciences, but I think there will be real opportunities to go beyond sort of the linked data that we produce ourselves and make use of other resources that are being added to it. And I think we have yet to really explore that potential, but hopefully we can start looking at that now that our data quality is improving. And so at the core of the project is really using a simple set of conventions and making use of Semantic Web technologies to provide linked data for the life sciences. So the hope is to reduce the time and effort that's involved in data integration and publishing, so that we can basically do a little bit more science instead of spending our time always, there's so many groups that would use common resources like OMIM or GenBank or whatever it is. So if we can standardize, I think, the production of these data, then it'll make it easier for everybody, and we can do more useful things, more complicated things. So the main features of Bio2RDF Release 2 is basically that what we've done is we've consolidated all the conversion scripts and we've updated them to reflect the sort of the latest in the data that's being produced, and so that our converters are more accurate. We have mapping files to basically move from the siloed data that it might be into mappings into other ontologies and terminologies to improve their semantic interoperability. And that all of this is open source and freely available in GitHub. And I think GitHub as a platform for software development is extremely promising. It allows people to not just clone the repository so that everything that we do is reproducible, but it also allows people to fork it and make improvements and submit patches and so on. So we're pretty excited about that potential and the potential for growth in new areas. So at the core is that Bio2RDF enables this data integration through both at least minimally a syntactic data integration where we use a common language, RDF, and we impose a common URI pattern which is regulated through a resource registry. So this Release 2 has 19 core datasets, and themselves, some of these are also aggregate datasets, so they contain many other data sets, and I think that's an improvement over previous where we're starting to use aggregator data sets. And all of these now have provenance and endpoints, and so they're resolvable entities, and we can look them up, we can query about them in the graphs. And we also have pre-computed graph summaries, which enable basically their graph summaries that allows you to know basic statistics about the data sets. But it can also be very useful for instance, for SPARQL autocomplete services and other lookup services. So the web application, which has been provided by Peter Ansel, basically allows for entity resolution, this query federation across both Bio2RDF endpoints, but then also enables us to query other people's endpoints and make them appear as part of the Bio2RDF network. So the Bio2RDF, our guidelines for RDFization are now published on the wiki at GitHub, and so the idea is to basically at least provide what's important in an RDFizer for Bio2RDF in order to interoperate into the network. And so these are just common, I guess, procedures that we would follow both in naming and in a kind of annotation that we would expect to see for any one entity, and also some instruction for the provenance. And these guidelines are basically built into a library that we developed for each of the scripts that are currently available for Release 2, and this is a PHP library. So we kind of enforce the guidelines through the program API. So the converters, like I said, are open source. They're available at GitHub. So every single one of our data sets, you can see the script there. And I think important is that because we've done this sort of first pass, we know that there's always room for improvement for these for some of these data sets, and I think that's sort of the opportunity. If you see something, if you see an error, then at least through GitHub now you can record the issue right away, and then we can assign developers to that. And so whether that's us or whether that's you because you want to contribute to the project, then I think that becomes possible so that your contributions are noted. So at the core, many of you have seen this before, but at the core we have a basic simple naming convention, and basically for any entity, the convention is that we use HTTP URIs, so HTTP followed by the domain name bio2rdf.org / the namespace, which is like a short name prefix for the database or the dataset, followed by a colon and the identifier. So for instance, for drug banks, IRI for leucovorin would be this bio2rdf.org / drugbank :db00650. So drugbank assigns this identifier for that named entity, and this is the name. This is the name that we give it in Bio2RDF. So obviously, all of these entities that we name are entities that you can look up when you put them in your browser. And I think that's part of the contract that we have when we want to provide data on linked open data, is that these named entities are resolvable. And moreover, so the interface that we have with the web app just basically uses Puppy. So this is fairly old, and we really want to update this, and we certainly will. But at least for now, it's basically just gives you the set of triples in this table format. But importantly is that all the links are there so that you can browse through the network and actually go from SPARQL endpoint to SPARQL endpoint through the dataset links that are provided there. So like I mentioned, one of the big developments in this release was the fact that not only do we create a common API, a PHP API, to enforce the generation of the triples, but also that the URIs that we generate are also validated. And so we do that by having a registry of the different namespaces that we know about in order to alleviate the potential for misspellings or variants of the same entity, but with a different prefix. And so all of these variant prefixes for a database are now recorded so that we can normalize it to one sort of standardized name. So that registry in Release 2 was implemented as part of the PHP API. But what we did in the summer is we knew that what we had to do is get that out from the script and into its sort of own common database. And so in the summer, we curated about 2,100 entries in a Google spreadsheet, which includes the data sets that we have seen in Bio2RDF, those that were in the Miriam database, those ontologies that are in BioPortal, references in Uniprot NCBI, and the nucleic acids research database issue. And so basically, we went through, we looked at, we tried to create descriptions for the data sets, identify the organization, the website, and provide the HTML resolution template for any named entity in those data sets. So we just basically have a de facto resolvable HTML page should there be one. We also added the description for the identifier syntax in as a regex, and provided licensing information when there was some, and also basic rights that came or was afforded by this license, at least how we interpreted it. So we're currently working with the identifiers.org team, which is Nick Juddy, Camille Leib, and Nicolas Lenevard, to basically merge this dataset registry that we created with the one from Miriam. And so that will afford us the potential that whenever you see an identifiers.org URI, you'll be able to navigate into Bio2RDF to see what we know about it or what links to it. And vice versa, that if you're in the Bio2RDF network or you want to query the Bio2RDF network with identifiers.org URIs, you'll be able to do so, even though our underlying data will be with Bio2RDF.org URIs. And so we'll have that sort of reciprocal functionality. And at the same time, that will be our main repository for our registry of namespaces. So how we coin our URIs will be specified in identifiers.org, so people can look them up in order to figure out how do I link to Bio2RDF. It will be obvious through the registry services what the URI is for some entity that you're interested in. And at the same time, what Miriam did very well was specify the syntax for the identifiers. So regex expressions. And so what we hope to do sort of in our next release is to make use of these regex patterns in order to validate any URI that we generate to make sure that it follows the syntax that we expect, and if not, throw an error. Okay, so there's a few big changes in terms of the naming. It was haphazard before, like at least we knew from a named entity, the syntax was pretty clear. But for those things that we created in order to RDFize the resource, then it was pretty haphazard. And so we've consolidated that into a few patterns. So the first one is for types and predicates, relationships that are generated in order to support the RDFization from, say, a text file, a tab file, flat file, XML file. Then we follow this notation where we have bio2RDF.org / the namespace _ vocabulary : and then some identifier. So that could be either could be a literal sort of something that you can look and see, but it could also be an MD5 hash of the label. So you have both of those cases that occur. But we have it then for the types and the predicates. So that's how you know that that vocabulary has been created by the parser in order to support the RDFization process. And then we have another namespace that we create, which is the namespace _ resource. And that one is for nodes that we create, again, in support of this RDFization process. So for instance, in drug bank, there are reported drug-drug interactions, but they have no identifiers. But we want to instantiate a type, which is the drug-drug interaction, and we need to give it an identifier. So the parser gets to choose how it's going to uniquely identify this. So in this case, the parser chose to have the first drug _ second drug, okay? And it's to point that this is a resource that we created to support the RDFization, and it's also not necessarily stable. So it can change in the future. For instance, drug bank may introduce their own identifiers, or we may have to create a more sophisticated, unique identifier for whatever reason. So what happens, so this is an example here of that, a portion of drug bank where we have the named entity. So we have drug bank DB00650 that has a label. The label also follows a particular syntax. You have basically the title followed by, in square brackets, the namespace : identifier. So that's sort of an expectation of our RDF advisors, is that they produce at least those, so that whenever we're doing some kind of search across all the resources, we can readily identify the source in which that label has come from. So there we have the typing to the drug. We have a relation between this drug and the drug-drug interaction, and you can see it uses the drug bank vocabulary. It's DD interactor in. So that relates the drug to the drug-drug interaction, and that also has a label. Okay, so this is basic schema, I guess, for Bio2RDF data across all these updated data sets. All right, so that's at least within one data set. Of course, we have relations that link entities from one data set to another data set. So here on this slide, we have drug bank and PharmGKB entries. And so you can see that the PharmGKB entry has a relation which basically points to the drug bank entity. So you can see that the relation that's used here is this XREF relation in the PharmGKB vocabulary. So that relates these two entities across these different data sets. And when you're navigating the linked data, you can move from basically one endpoint to another endpoint by following that. When you look up these entries, there's the option to find basically the links, what links to those entries. So for instance, if you go to a glucoverin entry, the drug bank glucoverin entry, there's a link at the top which is find intra-namespace links. And that basically says what links to this entry. And so when you click on that, you see that there are links from other drugs, from drug bank targets, and from PharmGKB. So inherent in the web app and the way that we prepare the web app is that we look at all the links between the namespaces so that we can query the appropriate endpoint in order to populate that. So we don't do we don't query every single endpoint. We already know what content is in those endpoints, what namespaces are being mentioned in those endpoints, and so which ones are potentially going to provide information. So this page basically federates those endpoints that we know the content. There's content in there that links to it. So what we have at the end is basically a network of linked data, and we know we have, every URI is basically now validated in the sense that we don't have the mistakes that we had before where we had misspellings or name variants of these URIs. All of these have been consolidated now, at least for the updated data sets. And so now you can reciprocally navigate through the linked data network in Bio2RDF. So we have, I wanted to, wait, I guess I didn't have a list of those data sets, did I? Or does that come later? I think maybe it comes later, so we'll hold on to that. So another big aspect was that we added provenance metadata for each of the data sets. And so at the core is that for any entities, for any named entity, there's a relation, which is the void in dataset relationship to link the entity to the data set. And from the data set, then it's a void data set. We have the date in which this data set was created. And you have to remember this is our RDF, the Bio2RDF RDF data set. So we have the date in which it was created, and so the parser populates these fields as it creates the linked data. It has a label, which is to indicate that it's this. So in this case, we have mesh data set, which is produced by Bio2RDF. We have a list of we have the license, if it's available. So that would be a pointer to the to a license document, although that's not, I think, fully integrated yet in many of these. But at least there are an indication of the rights that come from the Bio2RDF data set. And the important thing here for the rights, so for instance, we have the ability to use, to share, to modify, and by, basically it's a by attribution sort of license that we offer for our data sets. But the important thing is that there's a relationship between this data set and the source data set. And so you can go from this one to the other one through the W3C prov was derived from. But that core data set, so the original mesh data set, may have licensing terms that make our terms not available to you, right? So for instance, for mesh, the ability to redistribute the data is restricted. You need a license from them in order to do that kind of activity. So while our data has this licensing, it is incumbent on you to look at the original source license terms in order to determine what your rights and responsibilities are, which we may not be able to provide you in whole. There's a link to the creator, and the creator for our Bio2RDF datasets is the parser. And so you get a pointer directly to the GitHub parser, and the publisher then is Bio2RDF. And the mechanisms of the availability of this dataset would be through basically the SPARQL endpoint in which this dataset is published in, and as well as where you can get the RDF data, which is this dataset. So all of these comprise our current provenance model for Bio2RDF. And what we're interested in, I think, at this point, having seen the prior presentations, especially the OpenFacts one, is basically can we make sure that we basically are we're populating the fields that we can populate, and that basically the structure or the predicates that we use are in common with that of OpenFacts. So I guess we'll need to do a little bit of work to ensure that that's the case. But at its core, this is a pretty simple, straightforward provenance description for our Bio2RDF datasets. So the other thing that we provide now is these pre-computed metrics, and these are basically just metrics on the graph. And the idea was, what we noticed up until basically all this time, is that people tend to do these queries anyways to get an understanding of what's the content in our databases or in our data sets, our SPARQL endpoints. And these are pretty expensive queries sometimes, especially when you're asking to aggregate types over relations. So we basically do that now, and we publish that in a graph that becomes accessible to you. So here are the things that are in there: the number of subjects, predicates, objects, and unique types. And then we have combinations of predicate objects and predicate literal pairs and their frequencies. And then we have unique subject type, predicate object type links and their frequencies, and then subject type predicate literals and their frequencies. So I'll give you an example for, okay, well, first, like I said, all of these are stored in every endpoint in a particular graph. So the graph names are as follows: so bio2RDF.org/bio2RDF-namespace-statistics. And you can retrieve these using SPARQL, of course, pretty simply. But here's an example for the subject type, predicate object type counts for, which one is this? This is for drug bank. So you can see the first entry, you have the type is pharmaceutical. There are 11,512 instances of pharmaceuticals that are linked to 56 instances of unit through the predicate form. Okay, so the drug bank vocabulary : form is the predicate. So this basically tells you, if I have an instance of some type, how is it connected to other types of things? And so this is basically a nice graph summary, which can help you in your formulation of a potential query, right? These are all the possible paths you could follow in order to go from one entity to another entity that is typed, at least. So it doesn't include relations that go to resources that have no typing to them, however. Okay. So for instance, we can, just looking at the graph, you can say, okay, well, I'm interested in drug-drug interactions. And so I see that there's a relation between drug-drug interaction and drug, and the predicate is DDI-interactor-in. Okay, but you can see that this relation, right, it's not symmetric. So in order to access that, you have to put the query variable for the drug on the left-hand side of DD interactor in, and the drug-drug interaction on the right-hand side. So that can certainly help you formulate these queries, but thankfully people have already thought about this, and so there's this SPARQL ED tool, which is available for free, and it's open source from Syndice Tech, which can use this summarization graph to populate or to help you do autocomplete, context-sensitive autocompletes in your SPARQL queries. So that's a slightly different graph than the one that we produce. So we actually have these two graphs. We have our own statistics graph that's in the endpoint, and we also have this SPARQL ED graph in the endpoint. So if you want to use one of the endpoints, you can deploy the SPARQL ED tool on your own machine and simply point it to that graph, and then you'll be able to do the autocomplete functionality. And it works pretty good. So it's definitely a nice addition. Okay, so as you know, Bio2RDF covers, like many of the big biological databases, like Entree or NCBI Gene. We have PDB, we have KEGG, we have OMIM, and a bunch of others. And so these are the data sets that we have in Bio2RDF. We've updated the Affymetrix probe sets. We have the biomodels database from the EBI. We have the comparative toxicogenomics database, or CTD. We have drug bank and NCBI gene. We have the gene ontology annotations and the Hugo gene entries. We have homology interpro iPro class IREF index for protein-protein interactions. We have mesh, and we have a portion of BioPortal which conforms to the OBO part of BioPortal. And so it's our hope, since the NCBO BioPortal team is producing a SPARQL, or has produced a SPARQL endpoint, what we're going to probably be doing is basically, instead of trying to recreate that resource, is basically trying to tap into their existing SPARQL endpoint. So that's likely going to change in the future. We have the NDC, OMIM, the PharmGKB, SabioRK, which is a kinetic reaction database, SGD, and the NCBI taxonomy, which is included. Actually, yeah, no, we parse that separately, but it's also partly in BioPortal, not everything. So that's 19 updated data sets, and we have 1 billion triples that come out of that. And so we have some data sets that we have deprecated, and that's because they are found in some of these aggregate data sets like IREF index and Pathway Commons. So Pathway Commons was enlisted there. Again, Pathway Commons has also produced their own RDF. We loaded it into an endpoint, and we're just in the process of, I guess, creating some more linked data that's in that available in the BioPACS format, but is not readily accessible. But that's ongoing. There's a few big data sets that we still need to process, and that's GenBank, PubMed, and RefSeq. So we haven't yet updated those endpoints, but it's our hope to do so for Bio2RDF Release 3. And these are pretty big, so it takes a bit of time to process them properly. All right, so like I mentioned, a big difference now is that we have this PHP-based library, which acts as a point of integration. It basically provides a set of APIs to work with data files and to produce linked data that conforms to the Bio2RDF conventions. And it checks, it generates and checks these URIs against our dataset registry and helps you more easily generate the dataset provenance. So that's a nice sort of new addition. I think that even this library, we're planning to actually rewrite it to become a little bit more powerful from an object-oriented point of view, and also to use what we hope will be the registry produced between us or produced through identifiers.org. All right, so Bio2RDF, you can access it through downloads. You can get the data. So we have data dumps. We have these statistics files, and we also have the compiled Virtuoso database, which out of the box has the faceted browser up and running. You can use the web interface, so the web application, to do the lookup, and there's some services like these query federation services and lookup services across all the or any one of the endpoints. We have the SPARQL endpoints for each of the data sets, and you can use this SPARQL ED editor to help you compose queries over those endpoints. And each one, of course, because they're virtual, so you get the faceted browser that comes out of the box. And for what it's worth, I find this extremely useful to start to find out, well, what's in this endpoint, and to delve through it and start creating complicated queries without having to write any SPARQL. And it takes a little bit of time and effort, I guess, but not very much, to understand how this functions, how you can get these aggregated value counts for any portion of a query, and to be able to move from entity to entity and add more restrictions on them. But it's certainly a powerful tool, and it comes for free with the Virtuoso endpoints that we produced. Of course, there's third-party software. So, Aggregate is here, and he showed me last week a demo of using the Sentient Knowledge Explorer with Bio2RDF endpoints. So we're pretty excited about that, and we think that's definitely going to help increase the utility of our data, and also to enable people who might not know very much about SPARQL or the Semantic Web to compose fairly complicated queries in a graphical format. I think that's quite promising. And so what I'm hoping is that we'll be able to produce some tutorials or blog entries that indicate how one can download the software and start using it. I know that Eric is also going to be demonstrating the next version of the Knowledge Explorer at Seashell, so I'm pretty excited to see that as well. MetaOM has Distill Bio, which I'm not sure to what extent they use Bio2RDF. They certainly indicated to me in the past, and we've always been a little bit reluctant because of the quality issues we've had. But now I think is a good opportunity to revisit our endpoints and to make use of them, or to bring in that data and show functionality in these kinds of applications. FluidOps also showed me their information workbench when I was at ESWC last year, and so again, that was kind of interesting because it allows you to basically capture these SPARQL queries, semantic wiki kind of format, and I think that might be promising for us to demonstrate simple queries over the endpoints that we have. So one of the big issues, though, is that every one of these data sets is published with its own schema. And the challenge then is if you have data sets like drug, we have a few of these chemical drug data sets. And so how do you query across these endpoints without knowing that you have to explicitly create separate queries? And so that's a challenge of query federation, and that's basically data integration against these sort of distributed endpoints. And so wouldn't it be nice if we could just, formulate a query using one vocabulary, and it would automatically go and fetch all of these different data from different endpoints. So that's a goal that we want to realize, I think, in the next 1 to 2 years, and that we can automatically find these endpoints and create these simplified queries. So the challenge, however, is that each of these endpoints, each of the data sets, are published independently of every other data set. So what Bio2RDF does really good is just take data, whether it's in a flat file, XML format, text file, tab file, CSV file, whatever it is, and turn it into RDF. So that's at the core of what we do. But the challenge is that the patterns that we use to express these types and relationships do vary from one endpoint to another. And part of it could be just, in some cases, you don't. So for instance, you might have protein-protein interaction data, and it's simple to just have entity interacts with entity, right? And so it's a simple triple. But then you have others like IREF index, where you have actually an interaction where the interaction tells you that, yeah, it involves these two, but it's also, here's the experimental condition, here's the publication. And so you basically have this n-ary object. And so you have two different representations, and how do we move forward from that? Do we get one endpoint or one data set to be produced following the similar pattern or not? And so at this point, we think that the most important thing is to get it in RDF. Whatever the underlying structure might be is something that we can reconcile in the future. And I think more and more people see that as being, a sort of a fast and furious publication aspect. But then there has to be some integration that follows. I mean, the contrast is that, you have a standard for every data type, and everybody conforms to that and must implement their data. But that might be challenging, and it also requires people to cooperate, which we know can be difficult to do, and time-consuming as well. So we don't want people to slow down the publication of data, but on the other hand, we have to make sure at the end of the day that these data are interoperable and it's easy to query and find this information. So we know we have to do this. We have to do this semantic normalization step. But for us, we figured that it'll happen in a second step after the initial publication. And part of the strategy is to use ontology to do this, is that you have a common vocabulary with common patterns for expressing information, and that you try to conform to it. So the ontology we've been developing in the lab is the Semantic Science Integrated Ontology, or SIO. And so this is an OWL ontology for the representation of fairly diverse biomedical knowledge and artifacts. And so we're developing this sort of global schema for doing this. And part of what we've been doing then is trying to map the Bio2RDF types and relationships, at least, to the ontology that we have. And while that doesn't solve all the problems, it basically does some level of semantic normalization. Certainly at the type level, it's much easier now that we can express, give me all the SIO genes across the linked open data network that we have in Bio2RDF, and we can find those through the federation case. But it doesn't solve the problem that I indicated before with the protein-protein interaction, where you have structurally different representations of information. And so that's going to require something a little bit more sophisticated. Okay, so this project, we have this mapping project. Again, it's also in GitHub, where we map the types and relations in Bio2RDF to SIO at least. And potentially then that could be mapped to other vocabularies that are available out there, either by just linking to SIO appropriately, or to saying, well, we're going to do it slightly differently than SIO does, which is fine. So now what we're able to do, at least, is to do SPARQL 1.1 federated queries using SIO vocabulary across our different endpoints. And so here's an example where we fetch chemicals in CTD and proteins in SGD that basically participate in the same gene ontology process. And so we can look in these two endpoints, at least the CTD, the SGD endpoint, and of course, if we wanted to do some queries over the hierarchy of the gene ontology itself, we could query our bioportal endpoint and get different kinds of integration through that. All right, so to summarize, basically what we've done is we've consolidated and updated all our conversion source code, and it uses this enhanced PHP API, which makes use of our registry. And we have our simple design patterns that allow us to now regularize the representation of these data in our endpoints, in our datasets and endpoints. And I think that's nice. It's just aesthetically nice to see a pattern across all the datasets. So that's awesome. And we have some preliminary, but I think effective data provenance metrics available for quick querying of that content, and also provides us with an avenue for growth in the future, that we might be able to actually publish multiple different versions of the data, and you'll be able to know which one that you're looking at. And of course, as an open source project, and now that we've pushed everything to GitHub, I think there's the opportunity for people to actually work with us to improve the quality, to report problems that they may see, or and also to fix the source code itself, and so that in our next releases that we'd be able to produce that improved data. So we're hoping for twice yearly release schedule. So the next one is basically going to be at the end of June, and we're hoping that that will probably coincide with the biohackathon in Japan. And we hope to include these large-scale data sets, RefSeq, GenBank, PubMed, and the PDB, and to include some of the linking open drug data endpoints. So we have CIDR in a beta format. We've released OMIM. We have LinkCT also in beta. We know Kembl Campbell has just released another version, and they're planning to produce RDF. So they're in the works of doing this. So we hope that, in fact, the EBI with, so it's the GXA, the Gene Expression Atlas, you have Campbell, you have the biomodels, and I think there's a couple other data sets that they're planning to make part of EBI's RDF. And so if that becomes true, then we'll simply have Bio2RDF query those endpoints directly, provided that it makes sense for us to do that. We are already tapping into the Uniprot endpoint. So when you look up things in Bio2RDF, which are Uniprot entries, we actually query that endpoint and retrieve those for you on your behalf. But we're, again, hoping to sort of maybe think of how we might do that with BioPortal if it's possible. We have a project which is called Open BioCloud, which is with Dairy and Syndiche Tech, and that's to basically create a better interface to accessing the Bio2RDF data, to explore and navigate, and to share queries and other, interesting aspects of Bio2RDF. So that's on its way, and I'm hoping that, again, by the summertime, we have something that we can show everybody. And of course, like I mentioned, we really want to showcase this new improvements in Bio2RDF with third-party applications, not just to say, hey, here's what you can do with Bio2RDF, but also to, I think, encourage and create an environment where tool developers have nice use cases that they can demo with their software to the variety of different consumers and clients that they may have or that they want to see code. Okay, so with that, again, I just want to, the main developers here are Allison Callahan and Jose Cruz Toledo in my lab. And, but of course, Bio2RDF is an open source effort. And I think this demonstrates that what we've been able to do is take over an initial, that this was created initially by François Belot, Marc-Éric Nolin in Quebec, and Peter Ansel built the web app. And we've had contributions from other community members through this time, but that we've really been able to say, okay, well, let's try to fix some aspect of the problem. So I'm hopeful that other people who find this project interesting may pick a component that they want to improve and also make that contribution. Okay, and well, of course, we've had lots of input, I think, about linked open data and how to produce it and make sure that it sort of is getting better and better over time, certainly through this community, the HCLS community. So at this point, I think I'll just stop talking and be happy to take any questions that you might have.
Wow, thanks a lot, Michel. That's a great presentation. A lot of great work.
Thanks.
Are there any other, and I have some questions, but does anyone else have questions? Handy? Okay, well, I'll just jump ahead with my question then. I have a question about language, multiple languages, and if you have a path for Bio2RDF in terms of handling multiple language labels and so forth. It's much like we discussed earlier in Japan. As far as I can tell, there are essentially two different approaches taken to this. One is, I think, what I last remember with SNOMED, for instance, that there's a Spanish version of SNOMED and there's an English version of SNOMED, and they're treated like two different entire resources. But on the other hand, you might have data that is annotated, which isn't necessarily, so you're not using the entire terminology to annotate the data, but you provide multiple language labels such as Japanese and English. So do you have any comments on that, or do you have plans for that in the future for Bio2RDF, or is it really something that just hasn't really quite reached the data sets that you're dealing with?
Okay, yeah, very good question. So one thing to note is which is on our to-do list for Bio2RDF Release 3, is to enforce both language labels and also data types. And so one of the things that we don't consistently do in each of the scripts is that when we see some literal, we don't necessarily, if it's an annotation property, we don't label it with the language like English, which is the primary case in this. Or that if we have a number, that we type it as an integer, a float, a double, or whatever it is. So I think we're hoping to, when we do our second revisit of our scripts, that we enforce these patterns. But so far, all the content except for PubMed, I think, uses it's basically like a Unicode encoding, and it's all pretty much English. So that being said, I just had a conversation with Toshiaki, and I think one of the things that we want to do for the next biohackathon is to help or to make links between Bio2RDF and the databases that they have. And so in that case, like when you have Ali, for instance, and you have Japanese labels and things like that, there'll be the opportunity, I think, for us to either display that kind of content or to indicate whether there's certain language content in there. But that's something that, from our perspective, none of our data currently has anything other than English in it.
Okay, thanks.
Hi, Michelle, it's Atif. I just have a quick question, actually, regarding the very first stage where you actually do the syntactic matching. How do you validate or how do you calculate confidence into how good the matching is? And do you actually make use of that number or percentage or whatever that confidence level happens to be?
So I'm not totally sure. Whenever we have an identifier, like the identifier from the original data provider, that's what we use. And we don't create links. We don't create links between data sets. So we're not doing what some of the linking, like the linking open drug data effort did, which was to take these labels to try to create links between them. We don't do any of that. So we only represent links that were in the original data set. So if one data if one data set made a reference to another data set, then we just follow our pattern to create the right URI. But we currently have nothing in place to create new links that may be obvious. Like it might be that if you go from drug bank to, I think we did this with the Chinese traditional medicine, right, for LODD. And so you're trying to make the links because none exist in those original data sets and nobody's linking to them, then you have to look at the literals. And so we don't currently do that, but we are interested in extending the number of links that people create. And I think the way to do that is probably through the link set mechanism, where you can really say that there's a relationship between these two resources using this tool that I used, right? It's not in the original data itself.
Yeah, that makes sense. Thanks.
Are there any other questions? I have one more. It's about the metadata, and you probably remember these discussions about where to store it and how to find it and so forth. And I thought that the suggestion that David Wood had a few years ago seemed to be the best option, given no specification of where to put metadata about a graph. So if you have a data set that's stored in a graph, then you could put statements about the graph URI itself, or IRI, into the graph, right? But I'm not sure if you're actually doing that, or if you have a Bio2RDF sort of naming convention like what you've done with the statistics graph, right?
Yeah, so you're bang on there. That's one aspect that has not yet been, let's say, standardized, is the naming of your graph. So when you go to an endpoint, sometimes all the files that we process are loaded in the same graph, and it would be bio2RDF.org/graph/the dataset name prefix. In other cases, I've seen I've seen that each dataset is loaded in its own graph, and so we haven't yet decided how the graph structure for our datasets. And I think an important point of conversation is if it is the case that you will be associating this metadata with the graph, then it's going to have to be a graph which is distinct from a future graph, right? And which means that we will have to have some mapping in order for you to go from a standard graph name to the most recent one, or there's some relationship between that, and that's not yet been worked out. So I think part of the problem is because graphs themselves are not recursive, so I can't really say select from this graph, which might be the canonical graph name, but say that there's a more specific graph that it contains. So you should query those triples too. So the problem is more like although we can set out a convention, in practice, if you want to restrict your queries to a particular graph, then you would have to know the specific version of the graph that you want to query, which you'd have to store somewhere.
So I would expect that if you have, that you want the most current graph, that you just, when you don't supply a version number, you get that. Whereas if you want a specific version from the past, then you can supply that somehow in the...
Yeah, so I think it's not that we're averse to this. I think we want to do this, and we could potentially do it for the next one. But I think we'd have to have a little... I'm happy to have the conversation with the community as to what should we do, and what use cases does it support, and to make sure that everything that we need to do what we do now is supported. But I think it's totally feasible. We just haven't really thought about it yet.
Yeah, that's interesting that I hadn't thought of that, of course, statistics will change as the graph grows or changes with time, so that does create a different type of problem than what we were talking about earlier in terms of things like license don't tend to change, or descriptions of the data set usually stay fairly stable. But yeah, if the statistics are changing, then you don't want them to refer to the wrong version of the graph, of course.
Yeah.
I see that.
But I think it's definitely a conversation that we should have, and I think it would be a good idea for us to sort of provide a spec which basically indicates how we should not just manage provenance, right? So this conversation we can have with OpenFacts, but then how do we also store it along with these other statistics that we're generating? So sure, I'd be interested in looking at that. Any other questions?
Well, thanks a lot, Michel. This is it's really a bright look at the future, I think. So thanks for showing us this latest release of Bio2RDF. It's a pleasure.
Thanks for the opportunity to speak. And like I said, if you guys are out there and you're trying it and something doesn't work, please let us know. Report the issue on the GitHub issue list, ideally, so that we can record it and we can put a developer on it.
Michel, is there a startup guide for this? So if you want to try it out, or like some sample use cases that we can try out?
Well, just go to bio2RDF.org, and there's a few examples that you can pull from the front page, and then there's also a link to some documentation. But it's mostly, it doesn't really have, I think, the use cases, that we want to develop with our partners. And I think that's going to be something that we hope to have, is a list of, okay, well, you want to get some, let's say, the genes that are involved in some disease, or the drugs that treat a condition, or whatever it is. But we don't currently have any of that written down.
Okay.
Probably worth taking a look at SPARQL Ed, too, if you're just getting started.
Yeah.
I don't know. Does it sound like a good starting place, Michelle?
Well, that and the faceted browser. I mean, you
Yeah.
can just go to, so here, let me see if I can just share this current application. Okay, so you can see this. So here's from the front page. There are some examples or whatever, right? So that's just to see the triples and to follow through the network. But here, there's a link about Bio2RDF. You can get the list of all the data sets here. So if there's something you're interested in, you can, so here for IREF index, this goes to the faceted browse for some protein interaction, which didn't work, so maybe I won't do that.
Well, that gene isn't important at all.
Yeah. It's everywhere, so just ignore it.
The metrics are here too. They're already listed for everybody to see, and they're linkable too. So I think that's a good place to start, is this page, and you can navigate into one of the data sets that you're interested in and do a search on it. I don't know what else see here. Well, it's probably like piles of people not trying it, so it's going to slow down. Okay, anyway.
So just, I don't know if do you know about SPARQL bin from Michael Hausenblas? He's got it's a sort of a paste bin for SPARQL, and I don't know. It just I like the idea. I don't know. I haven't really thoroughly checked out the implementation, but I can imagine people sharing SPARQL queries via a sort of a mechanism that beautifies the SPARQL query and maybe eventually has SPARQL ed built into it and that sort of thing. But you could have a collection of Bio2RDF queries that you could sort of
absolutely.
Yeah,
it's definitely something. I mean, Eric has some use cases I think that he's been working on. So I think again the hope is to try to identify these things. And yeah, using SPARQL bin would be ideal. But with Open BioCloud, something that we're developing, we want it'll provide a mechanism by which to capture and describe those SPARQL queries. Yeah. Over.
You say Eric, you mean Eric Gombos? Or
yes.
Eric Prudimo? Okay.
Yes.
Yeah. Just checking.
Anyway, so there's stuff to play with. So let me know if it. Let me know what you think of it, and if there are problems, please do let us know.
Thanks again, Michelle.
My pleasure.
There's so much material in your presentation that we can't even. They will have to come back to some things, I think. So, but it's great to see everything growing and in all directions like this. So.
Yeah. Thank you.
All right. Bye.
Thanks.
Thanks.
Thank you.